My talk today is on COVID-19 contact tracing, therapeutics, and vaccine development. Well, as physicians, we're very good at reading case studies. So I'm going to start with a case study of COVID-19 transmission in the family cluster. The report came from China. This was about an extended family of 7, and within a few weeks, 6 out of 7 got sick with COVID-19. As you can see from the top, a couple, a wife and husband in their late 50s, visited a temple fair on January the 19th before the Chinese New Year. They returned their home and then joined by their daughter and daughter's family of 4, and they stayed together for about 4 days. They were all happy, had a great time, no problems. And then the daughter's family went back to join her husband's family of 3, the mother, father, and grandmother. As soon as the daughter's family left, the wife developed fever, headaches, and fatigue symptoms. She got tested. 6 days later, PCR was positive. So as soon as that was positive, this illustrates contact tracing. The local public officials was able to intervene and notify the families involved. The second family, the third family, they got quarantined, but unfortunately, it's too late. By then, almost all of them got infected, except for a 6-year-old. One of them ended up in the ICU. So this cluster transmission shows you a number of things. One, all of them transmitted disease during asymptomatic period, during incubation time. So the lack of symptom does not protect you from spreading the disease or getting infected. 2, contact tracing can be challenging because the tests we have, like Dr. Liu talked about, we don't have a rapid test. The test often has a turnaround time of a few days, particularly in the beginning. This was in January, as you know, the gene was cloned on January the 7th. It was published on the 10th. So this was actually fairly quick. They had a PCR test fairly quickly, but still not fast enough to give the result back. It was easy to contact trace within the family members because there are not too many of them, but it would be much harder for them to trace back to the temple. How many people went to the temple fair? And so you can see the challenging of human contact tracing. It's easier said than done. Well, we have new tools, that is genomic tracing. What is the difference between genomic tracing and human contact tracing? Human contact tracing is tracing the human interactions. The genomic tracing is tracing the viral evolution as the virus spread from one community to another. Natural evolutionary mutations occur, and these mutations will serve as fingerprints behind for scientists to determine where the virus came from and where it's going. Well, one example is this nice study published in bioRxiv in May that talked about the source of SARS-CoV-2 in Europe and in the US. Many of you may not know our local hospital, Providence in Everett, was the hospital that received the first patient in Washington State and the first patient in the US. That was a 30-some-year-old gentleman who came back from Wuhan on January the 15th, and within the few days he developed symptoms. He came to our medical center, actually just 1 floor below my endoscopy suite on a weekend. He got sent home and came back, got admitted. And we activated, our hospital had an Ebola unit, and that was activated. He was taken care of by robots. It was in the news media, wild coverage. And then it went on for 6 weeks, quiet in the Seattle market. Nobody got sick. We thought, wow, maybe we got it under control. Nothing is going to happen until it hits a nursing home called Life Care Center, and people just started dying one after another. That was devastating. We thought Seattle was going to become the epicenter in the US as Wuhan to China. And we have a local genomic epidemiologist at University of Washington, Trevor Befford. He was coming up with a theory he called it cryptic transmission. He thought the first case who came back from Wuhan spread the disease quietly without having anyone showing up symptoms, and eventually led to the outbreak in Washington state. Well, that didn't turn out to be true, luckily. Based on this later, more extensive genomic tracing study, that first case was so well managed. He did not spread the disease to anybody else. He came to a dead end, WA1, Washington #1, did not transmit that to anybody else. And there were some other cases, went to a few other patients, never took off. And that arm took off. So there was some parallel transmission that came over from Wuhan and spread to Washington state, and we had our local outbreak. So you can see the power of genomic tracing that could not be replaced by the old-fashioned human contact tracing. And other factors being monitored, as we talked about, you're doing genomic tracing, you're monitoring the genomic mutations. One notable mutation is called D614G. What does that mean? It is a mutation in a spike protein of SARS-CoV-2 at amino acid number 614, a change from aspartic acid to glycine. What's the significance of this? This mutation occurred sometime in Italy, in Europe, sometime in February. The predominant form initially was the D form, the wild type, the regional type. As you can see, gradually by the end of June or May, over 90% of the world strain is taken over by this G variant. And multiple studies in the lab have shown this variant strain is 10 times more infectious. And the New York East Coast infection came from Europe and Italy. They got this strain. So that biologically perhaps could explain in part the East Coast was hit much harder as compared to Seattle and Washington area. If you look at the California data, Southern California had more of the G variant compared to the D. That perhaps explains why at some point Southern California was hit harder than Northern California. So this was an interesting genomic tracing finding. Well, genomic tracing studies does not always solve mysteries. Sometimes they create mystery, as Dr. Lu alluded to. You can do sewage water or wastewater testing as a sentinel or precursor to find a local pandemic. And this was a Barcelona study from Spain. It was published in Med Archive in June. Not only they discovered the sewage water containing SARS-CoV-2 viral genes a month before their first case, but also they found a year ahead of time, in March 2019, 2 out of 5 viral genes were present. So this was a huge mystery. What happened on that day in Barcelona? Did this come from an animal? Did this come from a person who died and then disappeared? Or this was a visitor? Why did it just show up in one day? It looks like real because 2 genes of the viral genome was discovered. So that's a mystery created by the genomic tracing. As a physician, obviously, we spend more time in medical therapeutics, so I'm going to take you through some of the recent progress in the medical literature. There's so many drugs, and perhaps one of the most media attention any drug has gotten is the chloroquine and hydrochloroquine, not only because we have our President Trump promoting it and believing in it, and the Brazil president who got COVID-19 infection also said the drug helped him. Is this really a game changer? Well, let's see. In the lab, the medicine, the drug really looks great. This study was published in Cell Research from the Wuhan Institute of Virology. And they compared chloroquine with remdesivir. Obviously, this is a wonder drug. It's supposed to be a wonder drug in the lab. And the y-axis is the inhibition of viral invasion into cells. Viral E6 cells are monkey kidney cells. As you know, the kidney and GI tract expresses very high levels of viral receptor H2 genes. So these are good cell models to study the virus in vitro. And here, they did 3 experiments. They added the drug either at the entry of virus, the green one, or after the viral infection, or the full time before and after. By any of the three criteria, actually, chloroquine at a concentration of 10 micromolar beats remdesivir hands down. So when you look at the data, you will say, wow, who cares about remdesivir? Remdesivir, if we have an old, cheap drug that's easily available and the concentration on 10 micromolars are well within the safe range of dosage. So this obviously was fascinating, and unfortunately, whatever worked in lab does not always translate into human success. I don't have time to go through all the randomized controlled trials, but one it's done in the US and Canada shows hydroxychloroquine did not offer any survival benefit, did not reduce mortality, and did not improve symptoms. It did not show much of a difference with placebo. If anything, placebo might be slightly better, but not statistically significant. What it tells you here is not only there's no difference, we have to be very careful as physicians and patients. When you have a self-limited disease, that means the symptoms will just get better on their own in over 90% of the patients. It's very easy to draw a wrong conclusion. You can drink water. He said the water worked because he survived. You drank tea and tea worked. He smoked, the smoking worked. So wrong conclusions can be drawn very easily. You got to do randomized control trials to control all the potential confounding factors, so the picture will become more clear. Well, if the drug did not work therapeutically, what about prophylactically? Can we use the drug because it's so cheap, easily available? And if somebody had exposure, you know, your next door neighbor you shook hands with had COVID-19, you did not have any protection, can I just take hydroxychloroquine and wait for my test results? Would this protect me? Well, this study published in the New England Journal of Medicine specifically answers that question. It was done with a very good sample size, about 400 patients in each arm. And to spare you all the details, just look at the p-value. They are nowhere near anything significant. So not only hydroxychloroquine or chloroquine did not turn out to be therapeutically beneficial, prophylactically, it did not seems to be a useful drug either. Well, what about remdesivir? You know, in the lab, it kind of got beaten by chloroquine, but this is a drug in the Chinese social media. It was dubbed as, the Xiwan, people's hope. And did this turn out to be the case? Well, the first study answering this question came from China because China got hit first by COVID-19. This study was published in Lancet in late April. In terms of viral load, all of them come down with or without any treatment. So there's no difference. The remdesivir really did not make any shortening of viral shedding. And in terms of clinical improvement, you see the two lines are very much similar. So it was a very bad disappointment for Gilead, I guess. So the clinical studies just kept coming. And in mid and late May, a multinational randomized control study was published, and the drug was given for 10 days, and the sample size was pretty large, 500 each arm, treatment for 10 days. After the whole study was done, it was concluded there was no survival benefit. Mortality rate was not reduced, and is not statistically significant. In terms of symptomatic improvement, the disease course was shortened, not by much, from 15 days average, a median disease course to 11 days. So you save 4 days of misery for a pretty good cost. And a few days later, a third New England Journal of Medicine randomized controlled trial was published. This time, because the previous study said the 10-day course was somewhat useful, you really cannot use placebo to do a clinical trial ethically anymore. So what they did was they compared 5 days versus 10 days. The sample size was a little smaller because they just didn't see any improvement. The study was stopped, and with the conclusion of they could not conclude because there were no difference in terms of mortality, in terms of clinical improvement between the 5-day treatment arm and the 10-day treatment arm. One thing good came out of this study is at least we know 5 days is no less effective, if there was any effect, compared to the 10 days. So the NIH guidelines subsequently changed. If somebody is going to use this drug, you can just use for 5 days instead of 10 days. And now the drug available to treat one patient, now you can use for 2 patients. So make the drug more available. And as of yesterday, we got a brand new study just published in JAMA. This is a multinational, international study involving many prestigious clinical centers. They did 3 arms, 1-to-1 ratio. They did a 10-day remdesivir treatment versus 5-day versus conventional standard care. What's interesting is the 5-day treatment seemed to have better odds of clinical improvement, and yet the 10-day treatment was no better. So clearly there's something wrong here. I wouldn't believe this was a really truly clinically meaningful finding. And again, consistent with all previous findings, 28-day mortality were 1 to 2% in all 3 groups without any statistical significance. So clearly this study says no survival benefit. In terms of clinical improvement, perhaps yes, perhaps not. So their final conclusion is clinical significance unknown. So that's fresh off the press as of yesterday. You'll be surprised, all these antiviral therapies fail to make much of an improvement. There are no game changers or no magic drugs. An old drug, dexamethasone, which is steroids, it's not a drug used for antiviral therapy. It's controlling the immune response. That actually saved lives. It came from the recovery platform from University of Oxford in UK. They gave dexamethasone about 6 milligrams, which is equivalent to about 40 milligrams of prednisone a day. So it's modest dose for about 10 days or so. And for the properly selected patients, those who are critically ill on mechanical ventilation, they can reduce the mortality rate from 1.0 down to 0.64. That's 36% reduction. For patients who required oxygen supplements, 18% reduction. But if you're pretty mild, you're going to live anyway, there was no survival benefit, of course. So what's interesting here is the antiviral drugs didn't really make much of a difference. The immune suppression drugs seem to have worked better. So perhaps the virus is not killing us directly. It's the virus that triggered overwhelming, out of proportion immune response that's killing us. Now let's move on to vaccine development strategies. And so for development of vaccines, the first thing you need to do is you got to identify a pathogen antigen. Which one are you going to go after? I will give you an example of hepatitis B because that's what I'm most familiar with as a gastroenterologist. Hepatitis B has 3 antibodies: surface antibody, E antibody, and a core antibody. If you went after the wrong antigen to produce the antibody, you will be very, very disappointed, because if you raise the vaccine that caused core antibody production, that's useless. That antibody does not have any protection at all. If you use the antigen to produce E antibody, you will generate something that provides partial protection. Usually your viral load will come down, but it would not eliminate the viral infection. What you need is the surface antigen. So for SARS-CoV-2, we're very fortunate. The target antigen has been identified previously from SARS-CoV-1 and the MERS studies. This is the spike protein that contains the receptor binding domain. If you block this antigen, the viral protein, you can block the viral entry into the cells. So that saved a huge amount of time. So now you know which antigen to go after. How do you make your vaccinations? Traditionally, you have 2 methodologies, 2 approaches. One is inactivated viral vaccines. You cook it, you treat it with formalin or whatever to kill the virus, and then you give that dead virus to the humans, and that will come with the antigen to generate all kinds of polyclonal antibodies. This approach has been very successful for hepatitis A vaccination development and the old polio Salk vaccination. I think there's still a Salk institution in San Diego, and was very successful. So this one approach has a good track record, but it takes time. This traditional vaccine development may take over 10 years. The second approach is somewhat more modern. It's recombinant approach, recombinant protein-based vaccinations. So instead of grow the virus, kill it, and use the natural virus as the antigen, you do DNA recombinant construct, and you use the construct to express protein. You make purified protein and use that protein as the antigen for vaccination. And hepatitis B has been very successful using that approach. And there are many different ways to make recombinant protein. You can make it in E. coli, you can make it in yeast, and you can now, I heard that there are recent social media reports, our home school in Chengdu is using insects to produce the protein. But whatever that is, it's basically a recombinant protein you made from a DNA construct, and you make the protein outside the human body. That's the established technology. Now we're in a new territory. That is, instead of giving proteins, which are antigens into the human body, you give genetic codes encoded in either DNA or RNA into the human body, and you make the protein or you make the vaccine yourself. And it's like you buy a blueprint and you assemble it, the product yourself. It saves the manufacturers tons of time and investment and money. And also the production of antigen in your body might be more sustained. So you might get a better response and you add a much lower dose. So that's the good side. And the uncertain side, I wouldn't say bad side, is this is basically an approach similar to that of gene therapy. Basically, you're injecting genes into the human body, except you're not giving human genes, you're giving viral genes, and make your cells to make the viral product. And what if this production of viral protein cannot be stopped? It just kept going. And for the rest of your life, obviously, scientists are making their plans to make this transient, so it's not going to express the gene forever. And what if the DNA gets incorporated into your genomic DNA and it would not stop? And there are two possibilities that might happen if that were to occur. One is you keep producing antigens and you desensitize yourself, like allergy shots. You give the same antigen over and over again, your body will desensitize, will not respond anymore. If that's the case, it would be a disaster. When the real virus comes and you think, okay, I'm desensitized, I'm not responding. That's one possibility. Another possibility is just my speculation. And you produce all these foreign proteins constantly, you end up with some kind of long-term autoimmune disease. So a lot of uncertainties. So what's the difference of DNA vaccination and RNA? As you know, genes are encoded in human cells in DNA, and the DNA has to be transcribed into messenger RNA, and the messenger RNA will be translated into protein. So you can either give the DNA that will go through the steps, or you can give the messenger RNA that saves you one step. And I can show you a few studies of each of these vaccinations. So the first human trial came from China. It was published in Lancet. And to save some time, it's just going to get to the point. By 28 days, this vaccine, which is based on adenovirus vector, at low dose, middle dose, and high dose, produced very high levels of antibody if you measure the antibody by receptor binding domain assay. So the antigen is purified, receptor binding domain, you use ELISA. But if you do a real life test with live virus, you see the antibody positivity drop from 94% to 50%. Why is that? Why is real life not as effective as you measure in the lab using purified antigens? Well, perhaps this study published in the Proceedings of National Academy of Science answered that question fairly well. the receptor binding domain typically at rest is lying down, quote unquote, lying down for immune evasion. It's not exposed. The protein folding is protecting it from seeing the antibody, and that does not get recognized by the antibody. This receptor domain will only stand up when it's getting ready to bind to ACE2 receptor. So that's why real life effectiveness is not always predictable by lab assays. And the next vaccine came from University of Oxford in England. It's also an adenovirus-based vaccination. You give 2 doses, the prime dose, the first dose, the boost dose. At different times, actually, the antibody titer is very comparable to those you will measure in recovered patients. So those are the convalescent antibody titers in recovered patients. So this vaccine seems to be promising as well. And then you have the RNA vaccination was published. I was told not to use the drug pharmaceutical company name, so I will refrain from mentioning that. This study involved Kaiser here in the Seattle area, because we were the first area got hit. And as you can tell, when you use RNA, mRNA, you can use very little dosages of vaccination. Instead of proteins, you'll be measuring in milligrams. Here, you're measuring micrograms, even as little as 25 micrograms. They were able to produce antibody titer similar, if not above, the convalescent plasma in recovered patients. So very promising. And then a second RNA vaccination trial was published 10 days ago in Nature, and this was a multi-international study. And again, you notice they used very little doses, 10 to 100 micrograms of the vaccine. They were able to produce high titer of IgG levels above the human convalescent plasma. The oldest vaccine study came last, August the 13th, published in JAMA in China. So this was only inactivated vaccine I could find in human trials, and it was fairly effective. So far we have taken different approaches of generating antibodies by vaccination. They have variable side effects. They all seem to be effective, at least in the lab, measuring the antibody response. They have not been tested in the field to see if they actually reduce the severity of disease or reduce the risk of infection. I am cautiously optimistic, and we will come out with a good vaccine or a few good vaccines, but that obviously need to be field tested. To put everything together, I put in this summary slide. We're here talking about pandemic control. There are a number of key components we need to consider. One is source control, infection source control. 2 is blocking the transmission or the route of transmission. And 3 is mass immunization. So if you look at infection control, the source control, treating the sick patients as physicians is only a minor part of the big grand scheme. You do contact tracing that tracks human interactions and human migration. You do genomic tracing to track the viral evolution, mutation during spreading. You do quarantine or lockdown for individual or community asymptomatic contacts. You do isolation, use a different terminology when patients people got sick. That's called isolation, not quarantine. And we physicians would treat people who are much sicker and would not recover on their own for home resting. And obviously the treatment will involve pharmaceutical drugs, and so far we have not seen anything wonderful game changer drugs. They work to some degree, but none of them really changes dramatically except for this old steroids, which only one center published. We really like to see any confirmations of that. Blocking the route of transmission is also an important step. Social distancing, universal masking, all very important. My messages to young people out there ignoring all these rules of route of transmission is just think about sexually transmitted disease. The route of transmission is blocked by wearing a condom. If you have no problem wearing condom for sexually transmitted disease, HIV prevention, you should have no problem to wear a mask if you want to breathe in the respiratory disease pandemic. Now, the last, not least, is mass immunization. This can be achieved by herd immunity, which was proposed by the British politicians early on, but they abandoned that quickly because the human sacrifice is humongous. What's more meaningful and smart and scientific is vaccination. And obviously that's developing rapidly. Well, that's my half an hour talk. Thank you very much.